Thanks for having me. Yes, so I am currently, yes, in Colorado, and literally like days away from moving to Houston, where I'm going to start a lab down there. So I'm very excited about this move, because I go from a medical campus to university where there's going to be a teaching component. And one of the classes will be nucleic acid structure, function. So I'm very excited to be working even more closely with Jill and, Riju and the whole team at Eterna on the educational component of Eterna. So today, Jill asked me to talk about motifs. She was like, "Can you talk about 3D motifs?" I said, "Sure." And motifs, patterns, I may use that interchangeably. And so this is really about going beyond the GC and AU pairs, what we call Watson-Crick pairs, because they discovered this type of base pair, which is also in DNA, right? So you can view this as kind of like a primer into, tertiary structure of RNA, like Jill was introducing, and why motifs are important when you start thinking about 3D structures. So why is this important? So if, I like to put RNA in Google, see what comes out. And then this is what comes out, right? So you may get a slightly different version on your end. So you see a lot of things, structure, that's good. A lot of diagrams, cartoons, and mostly RNA looks like this, lonely molecule that's not DNA, because all of a sudden lost another strand, so maybe it's not as cool. It still shows like this wavy line with a bunch of colors on it. And maybe some folding, right, going on. But this is not the way we think about RNA. And when I say we, I mean RNA structural biologists. And another example, so I was happy during COVID, I could get a snapshot of that going on with, Fauci at the time, explaining to Biden, well, how does this mRNA vaccine work? And it shows that, one of these wavy blue lines again, and kind of, it's good, but at the same time, I'm kind of like turning green and being like, okay, it's a wavy blue line with 16 nucleotides. This is not the mRNA vaccine we're thinking about. And this is not what RNA really looks like. So what RNA really looks like is more something like this, right? We think of these very compact molecules, and here they're all colored from like one end to the end, one end to the other, right? Blue, rainbow colors, blue to red through all the colors. So they're very compact molecules. They have fascinating shapes, just like proteins. And that, we have way fewer of these structures than we have of proteins, because for the longest time, we didn't think there was anything cool about RNA. So really the challenge is, well, can we get better? Can we solve more of these structures? Can we get better at knowing how these RNAs fold up, not just in 2D, but in 3D, in space? So you can think of these RNAs, the very simple model on the top left here is like it's beads on a string, right? You have A, G, C, and U's, you have a sequence. It has a polarity, right? It goes in one direction, which I didn't, add here for simplification, because enzymes make RNA in the same direction. So that's fine. Stretches of A, G, C's, and U's. And then we know that, when we have these Watson-Crick pairs, G pairs with C's, 3 hydrogen bonds, A pairs with U, 2 hydrogen bonds. So we get structures maybe like this hairpin on the right, where there's like a stem part, a loop part. So we have, helices, and then something else. Right? And what is that something else doing? So if we dive a bit more deeply into the chemistry, this is very simplified chemistry here. So those beads, right, those nucleotides, those flavors, they all have the same, what's called the same backbone, same skeleton, right, with a phosphate group, some kind of sugar, and on that is attached one of these 4 bases, A, G, C's, and U. And then A and G's are kind of the same family. They're a bit bigger. C's and U's are a bit smaller. What's very particular about the chemistry involved here, which is directly what's impacting the folding and leads to all these structures that I just showed on the previous slide, is that these molecules, when they're in a sequence, so everything happens in water. Like your body is like at least 60% water. So all these RNAs, they're involved in a water-based environment. And water doesn't really like the chemical component that are on the bases. So those components are sort of stacking on themselves, the picture on the right. So each of these bases, A, G, C, U's, is going to stack on another base. So you can think of, like I'm showing here with like, sphere mode representation of every residue colored by type. And I think I used the Eterna code right. So all these bases are flat, they're planar, and so they stack like pancakes, like coins, and the sugar phosphate, that backbone, goes on the outside of that because that likes water. So that thing likes to see water, and then the bases who don't, see water for rough approximation, don't like to interact with water that much, except maybe on the edges. They stack, right? So basically, when you have an RNA sequence, and you see that wavy line in the Fauci presentation, this is not the most accurate way of showing that. It's convenient, it's simplified. Because what it's missing is that every base in an RNA is stacked on something else, whether it's another base right next to it, like shown here, right? Yellow, red in sequence, or it's another base from somewhere else in the sequence, leading to some long-range interaction, or it can be some amino acids or some ligands. We heard about riboswitches earlier. Situations like this. So Riju and I, we've worked on, solving plenty of RNA structures. This is one of them here that we both worked on, group 1 ribozyme. So from a protozoan, that's from like an organism that lives in pond, and it's the enzyme that basically gave Tom Cech its Nobel Prize, right, in the late 80s. So what I've done here on this, little movie that, I don't know if you see it playing. I don't see it playing. It's going, but it's not going.
I don't see it playing.
It's stuck. Yeah, I see the bar moving, but the movie is not playing. So anyway, it's fine. Not such a big deal. But this is like 400 of these nucleotides, right? So you have 400 A, U, G's, and C's. So I colored in black all the ones that make the Watson-Crick pair, A, U's, and G's, and C, that you're very good at predicting with Eterna, that we have very good rules for understanding. The ones in red are not in Watson-Crick pairs. And so as you see, even if it's not rotating, there's a lot of those, right? So the big question always when we go after solving these structures, like, what are these guys doing that are not in Watson-Crick pairs, right? So in a nutshell, to review up to this point, and that's going to be a bit wordy, but then you have the slide, all the key points are on this. So RNAs are compact, right? So they're not the limp, wavy line spaghetti. They form these regions, and they're kind of like bundles. I mean, I like to think of them as like bundles of stacks, right? You can see some, succession of stems and things, and then they organize themselves in space. So maybe you have crevices, binding sites, proteins can dock and all that. So they're not these, ladders with bubbles and paired and unpaired regions that we don't know what they do. This is convenient for the 2D, but it's missing on the reality of the 3D. So the other thing is when we have these ladders, and I'll show you another example later, we draw these residues with lines in between to indicate the pairs, and there's also this white space, right? Because we have the space to write the letters. But in reality, there is no white space. Like, this is all a continuous element, right? Because of the stacking. And so I mentioned that. So you visualize it on the left here. RNA structures, they rely on AU and GC, but also many of other interactions. And I'll show you some examples in the coming slides. And you already get a sense that it's not just the pairs that do Watson-Crick interactions that stack on one another in helices, like everybody stacks onto something else, like I mentioned earlier. So it's all of this, all of these interactions, like the pairing, the stacking, whether it's Watson-Crick, non-Watson-Crick, that helps stitch together this structure, because they're going to they don't bring together distal paths, but they keep them in spatial proximity to lead to these particular structures that we can resolve using the methods of structural biology, whether it's cross-electron microscopy, crystallography, NMR in some cases. So I have one example here that I want to highlight. It's very well known in the community. People have been working on for like decades. So on that same molecule, so 400 nucleotides, and I'm highlighting here still in black and red this domain where you can see a very long stack here, a band, and then a shorter stack, right? And then in spheres, I'm highlighting what's called this tetraloop receptor motif. So there's the loop at the end of that continuous domain here that docks into a helix on that side, right? So we have mostly non-Watson-Crick pairs in this, like mostly the red ones. And so, for example, as interactions that we have within this region, so if we compare to like the AU base pair that you're very familiar with when you're a player at Eterna, here we see some kind of AU where the U is not at all as it is in a Watson-Crick pair, but yet you still have some kind of interaction, like shown on the top right here. Or A's can interact with one another. So there's one, when that structure was first solved, people were really struck because they saw, oh, we have two A's, right, on the same strand. So not on, across from one another, but on the same strand, interacting kind of like side by side, which is shown here. And you can have also some A's pairing with G's. Highlighted at the bottom right. So you really, can think about RNA as like any residue. If you don't know anything, you see a sequence, any residue in that sequence can potentially interact with any other residue in that sequence. So we are pretty good at figuring out the Watson-Crick paired regions. The challenge is in between. And so as Riju mentioned in his intro, well, we have AI now that may help us with that. And I think him and I and many more are thinking, of ways to harness that, to be able to target this non-Watson-Crick region and make sense of that. So what's interesting about this, I'm highlighting this tetraloop receptor motif, and you have a different way of rendering it here, which more ball and stick type, so you can see what the bases, where the bases are in space a bit better than when you see this clump of spheres. And so you see three sticking out of that loop here, right, and going into the stem. This is a motif. I'll lead us to the definition of motifs. So brings two regions together, stitch them together. You can find the same thing in different RNA enzymes, so shown here, and sometimes even multiple times in the same molecule, where you're going to find, two different regions that are going to be stitched together by that same tetraloop receptor motif. So what's interesting is that because we have different examples in these different molecules, and people have looked at, oh, we can have different sequences even, right, of loops, so stem loop here interacting with a receptor, that still nonetheless adopt the same structure and support the same interaction patterns. So people have shown here the secondary structure, right, green with a boxed in blue shown up here in 3D, and then a different one here, and yet a different one there, and some others below, right? And then you superimpose the structures of all this. And you can see that, yeah, it's pretty much the same, even though the sequence is completely different, right? Or vastly different between the loop and the receptor. So different sequences can have the same 3D, can support the existence of the same motif. So this RNA motif or pattern, right? The definition is this, right? So it's an ensemble of ordered non-Watson-Crick base pairs, right? Because remember, it's mostly what's in between the Watson-Crick paired regions. So it's very modular. Like I showed you on the previous slide, you can have different combinations of AU, Gs, and Cs that are nonetheless going to give the same tertiary structure. The whole thing, so it's modular within, if you will, the nucleotides, the base pairs. But the whole thing itself is a module. Like you've seen it in different other molecules. So it can be used, taken out, basically plugged into something else and have the same purpose during RNA folding. So it's recurring, right? As a corollary of this modular property is that you find them in several RNAs. So here are some examples that we are really good now at recognizing because we've seen them in several examples of RNA structures and people have done mutations on them. So we understand the importance of some of the non-Watson quick pairing. And we even have shown on the 2Ds all these symbols with the squares, triangles, and open, closed. So this is a nomenclature that was brought forward like in the early 2000s to describe these types of interactions because we were, we fell short with just like the lines that we use for AUs and GCs. Like how are we gonna, be able to, we need some symbols, right, to show what these pairs are actually doing in 3D. So people who work with RNA structures, they're able to see AG with a square triangle and, figure out in their head, oh yeah, the A interacts with the G in such a way and so forth. So all these have been well characterized. You see them mostly non-Watson Crick pairs. Some introduce turns, right? Some open grooves. They're known protein binding sites. Ongoing question is always, have we identified all the motifs? Probably not. Have we identified all the variations of all the motifs? Even less. How many motifs are there, right? And so these are ongoing challenges in the field. An example here of why it's important to look at these non-Watson Crick paired regions is like here you have boxed in red the same set of 3 GAs, right? Types of GAs, non-Watson Crick. And in that case, well, you have a continuous stack on the left, right? Everything stacks on one on top of another. Whereas here, because of the organizations, next to that box, now you have a kink, right? Other base pairs here. So this introduces a well-defined bend into the stack, and I'm going to illustrate that further on this slide. So here's a typical example of this is a 2000, plus nucleotide RNA shown in the typical cartoon view with a silhouette showing stems and bubbles, right? Ladders, loops, bubbles. So I'm highlighting 2 regions here, H2, so 2 domains, if you will, H34, helix 34, and 7. And I blow up on the right here, so you can see, and I do the same where I color the Watson Crick regions in black, the non-Watson Crick in red. So you can see there's a bunch of red, right? It's not just black. And then it looks like, okay, some of these loops here, I mean, this would stack, right? Would be one stem with some internal bubble with a bunch of G's and A's, kind of the same here, right? There's maybe a continuous stack with a bunch of G's and A's. Then when we look at the structures of that, what we actually see is like, well, the G's and A's are not doing the same thing in those 2 regions that are just shown as bubbles on the 2D. So here we have on the left, this is actually a continuous stack, and I symbolize that with those light blue dashes between the bases, right? So you can see, yeah, I go from this Watson Crick region to that Watson Crick region through this red region that maintains the stacking. Whereas in that case, this is the kink turn that I showed you on the previous slide, where I have like one stem, right? And then this region introduces the kink. So it's a well-defined bend. So I have this one stack here, it kind of ends here, and then there's a new stack that goes there. So in reality, this thing shown flat as like continuous H7 stack is actually bent with a bend that we can predict. And even bases like this that are shown, that 820 on the structure, when you look at the structure, it's actually stacked on the base that's from somewhere else in the RNA. So there's basically no base left unstack, right? I think that's the message. And so people, because we have structures of these enzymes, I showed you the ribosome, so these 2 pieces on the previous slide, these are like huge RNAs. And we have plenty of structures, often of the same family of molecules, but that has been teaching us a lot about some of these motifs. So for example, that kink turn that we know creates that band, it's a pretty defined band. And it's also the binding site for this protein, L7AE. So people have done, okay, we can take, we can engineer a kink turn in between two Watson-Crick helices, if you will. And then upon addition of the protein, we should be able to generate a certain shape in 3D with a well-defined 60 angle between those two arms, if you will, around the kink turn. So people, and I put the reference down there, people have done that with these 3 stems. So putting 2 kink turns, right? And then 3 longer stems of 33 base pairs that they can, measure by some form of microscopy. And then upon addition of the protein, like, oh, we should be able to form this triangular shape. And this is in fact what they see. So you see on the left here, where without the protein, you kind of see a worm-like structure, the kind of like, okay, the RNA is able to adopt all kinds of conformations. But once we add the protein, then we get into these well-defined shapes, like triangular shapes as predicted by the model. And this is completely engineered, right, based on the knowledge we got from natural RNA sequences and looking at these motifs. And I'm showing another example here of a triangular one, also a movie that's not playing. But because this one has a structure that's actually been solved. And here they use a different kind of motif called that PX motif, where it allows you basically to go from one region to another with the same strand and vice versa. So it's like a cross strand stack, if you will. So different kinds of structures that people are able to engineer because they understand the rules of Watson-Crick base pair formation and the rule of formation of these motifs, which are mostly non-Watson-Crick based. So maybe one day, right, we'll have something like that, where instead of, a wavy line, and we'll be getting some RNA nanoparticles. People are working on that for maybe immune system simulation, right? Drug delivery. You could imagine RNA nanoparticles, if you can organize them in such a way, you can have some sites that are, we know exactly where the sites are. They can be functionalized by binding small molecules. They can be small molecules or other deliverables inside these nanoparticles. So this, might be where we're going, at least one potential avenue. So thanks for listening, and I'm happy to answer questions you may have.